So our next speaker is Dr. Matthew Vander Heiden, and he's a professor of molecular biology at MIT and the director of the Koch Institute. And he's a practicing oncologist and instructor, and he has won so many awards, so I'm not gonna read them all. But he's a member of National Academy of Medicine, so please welcome him, and the title of his talk is Impact of Diet and Systemic Metabolism on Cancer Progression. Thank you.
All right, thank you so much, Jose, and thank you for being here. And I guess since I'm an organizer, I'm not going to thank myself, but I will thank Allison and Omer for all the hard work pulling together the symposium. As you might guess, they did a disproportionate amount of the work. Okay, here are my disclosures. Okay, so as you heard in the introduction, I still try to see patients, although do it quite in a small thing. But given that my long-standing interest has been in metabolism, as you might imagine, one of the most common questions I get from patients is, what should I eat? I probably get an email once a month, usually more frequently. It's always a terrible, sad situation, and it's largely about, should I try this diet? Should I try that diet? They often quote various people, sometimes some of the other speakers here, and their work. And I have to say, I always struggle with these questions because it's a really hard thing to answer, right? I think you heard the complexity of how to interpret diet from both of our first two speakers. I think we're going to hear it through the rest of the talks today, and I think it's quite important to really try to understand mechanism and resist the urge to try to give dietary recommendations from one thing or from one model system and extrapolate it to absolutely everything. But nonetheless, one thing that we've been thinking about quite a bit in my lab is really how can we understand sort of metabolic adaptations for all stages of cancer progression. And I think increasingly one thing that we've been very interested in is this notion that maybe some of these metabolic adaptations might actually be telling us something about other events that happen within cancer. And hopefully what I can share with you today is a couple different stories that both relate to diet, although diet in an unexpected way, about how our efforts try to understand these interrelationships have at least told us something about ways that diet might influence cancer. Now, I have to give a shout out to Evan Lien, a former postdoc in the lab, who a number of years ago really sort of got us thinking more about diet and really thinking about how could we understand dietary mechanisms with respect to the way cancer might develop. And he sort of came up with this paradigm, and we heard this mentioned earlier in the earlier talks about how what we eat at some level influences what nutrients are available to the tumor. Now, by no means is this a one-to-one relationship, but this sort of fit very well with ongoing efforts in our lab to really try to understand, well, what nutrients are actually present within a tumor and how might they constrain the ability of cancer to develop in different ways. And so I have to give a shout out to Alex Muir and Mark Sullivan, two former former graduate student, who really sort of set us on a path to try to how to think about this, where they developed quantitative mass spec techniques to sort of profile what is actually in the nutrient environment of different tissues, different tumors. And here's an experiment that they did way back in 2019, or published in 2019, showing that what we feed mice actually influences at least what nutrients are found in different tissues. And so for a number of years, many people in my lab are in the audience, and so many of you have contributed to work. I'm showing here an experiment that was done by Keen Abbott, a former graduate student, that I think was quite informative, where he basically took apart a mouse and basically isolated interstitial fluid from as many organs as he could get. He looked at blood. And sort of one big thing we learned from these studies is it turns out every single organ in our body seems to have its own unique nutrient environment, which has obvious implications for how you might think about cancers either arising or metastasizing to those different organs. But one thing that question that Keen asked, and I have to say I was obsessed with this question as well, is that, well, this is all well and good. It's really interesting that each of these environments is different, but what makes them different? And one of the largest drivers of difference across those environments actually turns out to be nucleotides. So this is not something you typically think about as varying all that much in the diet, but it turns out different tissues in our body have different levels of, some have higher purines, some have higher pyrimidines, different precursors there. And since many other work in the lab had been converging on nucleotide metabolism, this got us thinking about, well, how could we really try to understand this variation in nucleotides across different tissues within the body? And so I want to tell you about some work that was led by Rayan El-Bashir, a graduate student in the lab, who really wanted to think about this. And we ended up focusing on a leukemia model, and it turns out there's a variety of reasons why we chose to do this. But one is we thought how we could model different environments. So as I showed on or alluded to on the previous slide, what's in the blood is not necessarily what's in the tissues, but it turns out bone marrow as a tissue looks most like blood, and leukemia grows in blood and bone marrow. And so that sort of gave us a way that we could sort of leverage what might be happening across both human and mouse samples to look at this. This points out that the nutrients that exist across different BALL subtypes that we were able to get from Jana Pickman, this is blood, but bone marrow would look very similar, is largely, although not identical to healthy. And when Rayan and Keen worked to either use a mouse model of BALL that we got with the help of Mike Heeman's lab, our colleague here at the Koch Institute, or looking at blood or bone marrow plasma from children from Children's Hospital that Jana was able to get us, what we found is that the way that blood or bone marrow differed from animals or humans with BALL versus healthy control, many of the differences largely had to do with different nucleic acids. Now, the clinicians will say, oh, this is tumor lysis and stuff like that, and maybe that's part of it. But nonetheless, this ended up being an interesting system that we could think about, oh, do these differences in nucleotides across different compartments matter? And so the way that Rayan decided to tackle this is she said, well, rather than grow cells the way we've always grown them in RPMI, maybe we could reformulate them into media that looks most like the blood. And so she made what we call here MPM, that's mouse plasma-like media. It's basically media that contains the nutrients, not that we typically grow our cells in, but that match what is actually found in this case, the plasma of the mice with BALL. And I just want to point out that you can grow cells. You don't have to grow them in RPMI or DMEM. They grow just fine in lots of different medias. Turns out blood-like medias support proliferation quite well, almost identical. But if you just do metabolomics on the cells, remember, RPMI, so RPMI, we grow cells. There's no nucleotide precursors. MPM, we put the nucleotide precursors in that we measured in the mice with leukemia. And then if you actually measure, it turns out the biggest changes in metabolism within the cell, again, now have to do with nucleotides. And so Rayan then said, well, this is interesting. So big difference. This has nucleotides to salvage. This does not. So obviously these have to synthesize nucleotides. These have a choice to do either one. So then she wanted to ask the question, well, do the sources of nucleotides actually matter between these two different conditions? And so she did some fancy tracing techniques that I'm not going to bore you with, although I'd be perfectly happy to describe how we did this experiment during the question and answer. If people are interested in, she, not me, but she has helpfully color-coded here exactly what we found in terms of red is basically things that are consistent with salvage. Purple is things that are consistent with de novo synthesis. And not surprisingly, if you grow cells in RPMI, because there's nothing to salvage, almost everything comes from de novo synthesis. And maybe also not surprisingly, if you grow cells with nucleotides to salvage, they will salvage the nucleotides that you give them, okay? Nucleotide synthesis is expensive. Lots of people have seen this. But here was somewhat of a surprise, and that is we could also modulate the nucleotides in these conditions. That is, we could either add them to standard media, made zero difference in proliferation, or take them away from our blood-like media, and it made a big difference in proliferation. And for me, at least, this was a big surprise, because this is not nutrient restriction. This is nutrient different. There's plenty of plenty of nutrients there. But yet, there seems to be more of a dependence on salvaging those nucleotides, even though there's plenty of nutrients otherwise to go around to actually make the nucleotides that are present. In other words, nucleotide synthesis seems to be constrained in these conditions and not those conditions. And we could do basically experiments here to show this. If we knock out a synthesis enzyme, well, you don't do very well in standard media, but it turns out you also don't do well in plasma-like media, whereas you knock out salvage, you don't do very well in plasma-like media because somehow synthesis is constrained. So then we set out to find out what was actually constraining synthesis, and I have to say we first considered it would be all the macronutrients that are necessary to contribute to nucleotides. However, what we found was, and this is just metabolomics that is about as black and white an experiment as you're ever going to get. So the relevant situation here is the MPM minus nucleotides, and you see that in that condition alone, you accumulate GAR, ICAR, DUMP, you have lower levels of DTMP. If you know metabolism, you took 705 from me in this exact classroom, you know that these are basically the things upstream of all the folate utilizing steps in biology, which really says, this really suggests that folates are actually the issue, a micronutrient, not the macronutrient we initially thought. And we could prove this because it turns out the folic acid concentration is very different between MPM and RPMI. 2.27 micromolar in RPMI, someone else formulated that. MPM, we had 150 nanomolar based on what we measured. And if you simply raise the folate concentration in MPM to match RPMI, this entire nucleotide phenotype goes away. And so it's something non-physiological about the folate concentration that seems to be driving this phenotype. Now I want to talk about, well, that actually raises an interesting question, because what's actually physiological? Well, it turns out that folates, diet matters for folates. It turns out mouse chow has much higher folic acid than what humans typically consume. And if you look at, actually, this is the physiological range. It turns out we formulated MPM actually at the top of the mouse physiological range, which is higher, it's interesting, it's lab mice, than the human physiological range. And I'll just point out, here's a typical folate for a young female graduate student, Rayanne, who did the work, is 31 nanomolar. Okay, so very different, even much lower. And you can see if we titrate folates over the range, this will actually control how fast cells grow in this MPM condition. So I want to say, why is it higher in mice? Well, it's because these are lab mice. These are not mice from the wild. And it turns out we supplement mouse chow that we feed our mice in a facility. We give them, quote unquote, breeding chow, which has high levels of folic acid, which is why this is the case. And that's largely because of maybe the greatest public health intervention, or most successful public health intervention, which was done a number of years ago, which was basically supplementing folic acid within bread, cereal products that we give people, which dramatically has decreased neural tube defects. But I want to point out there's also a long history. We heard from Professor Willett this morning about folic acid and its inverse relationship with colon cancer risk. But I'll also point out that this was actually how antifolates actually developed as one of the first chemotherapies to begin with, had to do with an observation from Farber that giving folic acid actually accelerated leukemia. An interesting story we can talk about later. But it turns out we can largely repeat this. If we modulate folates in the diet and get rid of salvage, we can largely control how fast it is that leukemia progresses, and has an impact also on survival of the mice. And so I think this sort of pointed out for us that not just macronutrient composition of the diet, but micronutrient composition of the diet, which of course epi studies have found as well, can have influence on cancer. But I want to think about, so why might you get different effects of folates on either tumor initiation or tumor progression? And I think an interesting thing really comes from, well, why is constraining nucleotide synthesis a problem? I think what this says is that at baseline, nucleotide synthesis is largely constrained. But I think this becomes a problem because if you end up with imbalances in the nucleotides you have, it turns out our data says this doesn't matter so much for the RNA fates of those nucleotides. Those are quite high in cells. But where cells get into trouble is if they have imbalances in the deoxynucleotides needed to replicate their DNA, which leads to a well-described response known as DNA replication stress, ATR check1 signaling. Don't have time to get into this. But it turns out this is what happens. This is what happens when you have this constrained proliferation under situations where you have nucleotide problems because synthesis is constrained and there's nothing to salvage. That is, these cells experience replication stress, as pointed out by phospho-Chek1 staining. Now, if this is bad enough, this can, of course, lead to DNA damage. But I think the more interesting fate of this actually has to do with a finding that Peggy Sue and Brian Doe made a number of years ago, that this can actually influence gene expression state. I don't have time to get into this because I want to talk about a second story separate from this. But this sort of pointed out to us that, well, maybe part of how things like micronutrients or other things that might influence nucleotides contribute to cancer progression has to do with the situation that if there's actually mismatches and constraints that lead to DNA replication stress, this could be one of the drivers of heterogeneity in gene expression state that we know have to cooperate with the mutational accumulation that's necessary for cancer and is very important for things not just involving initiation, but certainly other factors like metastasis. Now, I want to pivot in my remaining time here to talk about a second story, which also has to do with micronutrients in the diet and really focuses on a very downstream effect of tumor, and that's sort of the interaction between tumor and host and this notion of tissue wasting. Now, this is work that we've been doing for a long time with Brian Wolpin's lab. It actually started a long time ago with Jared Mayers. The mouse data I'm showing is done by Yedish Gultekin, largely recapitulating what other people have found is that I think everyone sort of has the sense that pancreas cancer leads to tissue wasting. It's the most cachectic of the various cancers. And work that initially we had done in mice and later was corroborated in humans by Brian Wolpin, who we've heard mentioned earlier, and Michael Rosenthal, is that as cancer develops in a mouse, this is a mouse model, I could show you any number of ones here, that as tumors grow in the pancreas, you actually get wasting, in this case of muscle. And what Brian and Michael Rosenthal have shown is that this wasting can actually occur prior to diagnosis, that this actually starts quite early in disease progression. And so we've been interested in what's really driving these changes, and that's a whole separate talk. But Tony Zhang, when he joined my lab, had a different question, and he had an interest in muscle wasting. He trained in Eric Olson's lab at UT Southwestern and long had an interest in sort of muscle physiology. And he made somewhat of a surprising observation, initially in mice, but again with Brian's group and Catherine O'Connor, one of his medical students, we were able to corroborate this in humans. It turns out, in addition to the muscle wasting, you also seem to be developing an anemia. Now, this is not florid anemia. This is subclinical anemia. It turns out, if you talk to most clinicians about this, they're like, oh, this is all treatment related. But these are undiagnosed patients. This is at diagnosis. No one here, and certainly we didn't treat the mice, but even within humans, you can see that there's evidence of, in this case, showing hemoglobin being lower in the windows prior to diagnosis. And why Tony was interested in this was because of some work he did with Puneeth Iyengar at Memorial Sloan Kettering that actually argued that if you had cachexia, and that's defined mainly by weight loss, not maybe the best way to do it, you were much more likely to also show anemia within a larger cohort. And so what is actually driving the anemia here? So I'm just going to get right to the punchline. Tony did a bunch of work looking at it. And it turns out the anemia is most consistent with iron deficient like anemia. I'll get with that in a second. Low total iron binding capacity, low iron in the blood. And iron is at least contributing to this because if we give now a different micronutrient, iron, we can fix the anemia within the mice with PDAC. Now, I have to say that this anemia, it's not straight iron deficient anemia. It's not that they can't absorb iron. It's actually quite a complex phenotype. It's actually much more, the clinicians, it's much more a anemia of chronic disease-like thing, which looks like iron deficient anemia. It's not supposed to be corrected by iron. Some of that may be how we're actually able to manipulate the mice. Anemia of chronic disease driven by high hepcidin. So this is clearly something we see in the mouse models. But this is actually a whole body redistribution of iron that actually seems to be driving this. And I'll point out that iron is accumulating within the pancreas itself, also accumulating within muscle and fat, which are the two tissues that are actually undergo wasting. And I'll just shout out that Christine Chiao, some of you who may have heard her speak about this, I'm not sure it's on bioRxiv or not, but she's been presenting it for a couple of years, has a nice story about how high iron and white adipose tissue might be driving wasting. And of course, Tony, being interested in muscle, has really focused on it at the level of the muscle. And I'll point out that even if still don't know how Tony was able to pull this off, but we have muscle biopsies from humans with pancreas cancer who are either more cachectic or not, and there is higher iron in the muscle of the patients who are cachectic, and because we can do interventions in the mice, I can tell you that if you give a high iron diet, you will actually raise iron in the blood, and this actually, if anything, will worsen the cachexia and the body wasting that we see. Still trying to figure this out, but it turns out this high iron diet, while it fixes the anemia, actually makes, if anything, the cachexia worse and worsens survival of the mice, which is also, I think, a really important thing. And I want to come back to the first thing that I started with, which was this notion of how do I actually answer the questions when patients come to me and say this? Because, of course, you don't want to be anemic, right? And so the easy answer is, oh, you should take iron supplements because that will help your anemia. Or you should take folic acid because, I don't know, that will help your anemia for other reasons if you happen to be anemic. Although the flip side is the folic acid, could make it worse, although it may protect you from colon cancer, right? So number one, we have to think about what disease are we actually worried about preventing or exacerbating. That is, the context matters a lot. And also, we have to realize that some of our dietary interventions, where they may be good for a complication, like an anemia complication or a cachexia complication, may not necessarily fix the ultimate outcome. And of course, in humans, this is even more complicated because I haven't even talked about how this would interact with therapy. And if we believe our therapies work, which sometimes people argue with me, but I actually think our therapies do work, there's good evidence for that. Our therapies also and their interactions with that are something we also have to be careful of extrapolating here. And so lots of complexities in this topic. I'm really excited for the rest of the symposium. I'll just close by thanking you guys all for coming. I'll thank the people again who did the work. I have amazing people in my lab. I tried to get all of them up here, current members of the lab, including my army of undergraduates, past people from the lab who contributed to some of the work I presented, collaborators who contributed to the work I presented, our funding sources, at least as they exist this month. And I'm happy to take any questions, but I'm also happy to make sure that we save some time for the coffee break.
Yeah, we have time for 2 or 3 questions.
Okay.
So thank you.
Thank you for your great talk. So you just mentioned the folate pathways and nucleotide-related pathways are affiliated with this process. So do you observe the spermidine pathway also affected by this kind of pathways perturbations? And so as a result, the new genome instability will be affected.
Yeah, so you raised a good question. So genome instability, I think other metabolic, so certainly if you have nucleotide problems that lead to replication stress that's unresolved, that can lead to double-strand breaks. That's well described from molecular biology literature. Is that a driver of genome instability changes? I'm not sure. It may contribute somewhat to mutation. Even that would be somewhat controversial if you go to things like sort of these mutational signatures and how they do that. I'm not sure that lines up perfectly there. There may be other metabolic things that lead to genome instability, but that's a different talk.
Thank you.
Thank you very much for
Yeah.
sharing your work. So my question is for the iron. So the data are from mouse model?
Well, there's a mix of data I showed you from mouse and humans, right? So we don't have data. It turns out trying to get reliable data on iron supplementation in humans outside of a prospective study is really hard because it turns out, yes, you can look back at health records, and we actually had a call yesterday with a large health group about could we actually do some kind of retrospective analysis to actually look at this. I think it's going to be confounded, though, because there's patients who get prescribed iron by their doctor, and that's going to clearly be in the medical record. But if I ask out there, I'm not going to do it, but if I did a poll of people out there, I bet many of you take a multivitamin or an iron supplement, right? Especially younger women who can become iron deficient otherwise. So I think it's hard because this is an over-the-counter thing, and I think we need to go back to things like the health questionnaires and maybe the tools that we heard about in the very first talk, if we ever want to answer that question right, or do it prospectively. That's the other way to do it. But if it's early disease, it's hard to do prospective studies.
Yeah. So I think one of the last slides you showed about iron, is that iron level in the plasma or iron supplement? Like a
that
supplement.
was iron level in the plasma. So if you raise iron level in the plasma,
yeah.
That seems, if anything, and you give mice, this is now mouse studies, intervention studies in mice, if anything, you make the cachexia worse and the survival worse.
Yeah.
But again, now you have to, in humans, be careful extrapolating to humans, because now we're not treating the mice, we're not doing others. So.
Yeah. Okay, thank you.
Yep.
Thank you, everyone.
Okay.